Hi everyone, I'm talking about inselbergs and how they influence biodiversity within the Free State grasslands of South Africa. So just quickly introduction background about what inselbergs is and all that stuff, what I sampled, how I sampled it, then what we found, and then discussion and conclusion as every presentation goes. So firstly, let's just look at the broader sense of grasslands in South Africa, where they are quite unique and very, very important for human development in agricultural, mining, and urban development as well. And they are extremely diverse. We have around 6 or 7 large mammal species that's endemic to grasslands of South Africa, and that's a trend you can see around the world as well. However, these grasslands is quite homogeneous in their landscape, especially if you drive past Welkom in that area. It's just miles and miles away, or you can see only grasses. You sit there on your stoop and you look at how your dog runs away for 2 weeks and you just see grasses all the way. So these inselbergs bring a good form of heterogeneity into those grassland systems. So now's the question, what is an inselberg? So you have the geological definition, which is this isolated rocky outcrops, which is usually formed out of granite, or in South Africa it's dolerite. However, there's a biological definition as well. As these inselbergs erode, their lithology, but basically the soil can move downwards and down the slopes, and the vegetation, the inselberg and the rocky outcrop can influence, due to water flow and lithology of the soil, the vegetation around the inselberg. So the biological definition and borders of the inselbergs might be quite big or larger than the geological and topographical borders. So these inselbergs are seen as isolated rocky outcrops, as I said, and I'm going to focus then on the biological definition for inselbergs in this study. These inselbergs differ quite largely with the surrounding areas. As you'll find throughout the Free State, these inselbergs have a lot more woody vegetation than the surrounding area. And very interesting enough, you'll find more woody vegetation on the southern slopes than on the northern slopes, as you can see in this picture here. Where the red dotted lines are the ridges, and then the green side of the inselberg is your southern side, and the not so green side is the northern side. So these differences in microhabitat and microclimate then provide a space for niche differentiation, as Renee talked about earlier. And then this gives you a source of heterogeneity in this otherwise homogeneous grassland landscape. So then, with this heterogeneity, here you can see again the difference in the northern and southern slopes, where the southern slope is a lot more darker green of woody vegetation than its northern slope. And these inselbergs are quite also important in a different sense, as they are less used for agricultural management or for agricultural use, as you can't plant on them, and you don't really want your cattle to walk on these rocky outcrops where they are more in danger of breaking a leg or something like that. So with this heterogeneity, we can find that we'll find different species of insects and insects, small mammals, and plant vegetation on these inselbergs than in surrounding areas, as I previously stated, which brings to the question that these inselbergs might act like small little islands in this matrix of grass, in the sea of grass. However, this has not been fully studied or documented well on insects, and that's why my study will be focusing on insects and not on vegetation. There has been a few studies on vegetation where they found that the further up you go on these inselbergs, the more distinct the vegetation becomes from the surrounding areas. So, for my study, we looked at the gamma diversity, which is the overall diversity around the entire study area, so that would be including the inselbergs and without the inselbergs. We will also be looking at alpha diversity, which is your local diversity at each site. And then we'll also be looking at beta diversity, which is the turnover, the change in species between these different sites. So how did we sample these inselbergs? So we firstly identified the inselberg, and then we identified the summit of the inselberg. And then we sampled with sweep net samplings on 5 different sites over 8 inselbergs. We first sampled the northern slope, and the northern plain and the southern plain, as well as the northern and southern slopes, as well as the summit of each inselberg. So on each inselberg we then did 20 sweep net transects in one direction and back, so in total 40 transects or 40 sweeps, and then we did it the same in the other direction, as can be seen here. And we did this for 3 sampling events in the wet season, last year in the wet season at Bankfontein. We also used pitfall traps, as can be seen on all 5 transect types, as we can see there, where we put out pitfalls in each meter. However, this was only done for 1 sampling event, as rain rain smoothed out these pitfalls the other 2 sampling events. Then we went to the lab and we identified 15,000 individual insects to morpho-species level. First to family level, and then we divided them into morpho-species, where we found 322 different morpho-species across 8 inselbergs, across 40 different transects. And then we calculated rarefaction curves to see if we did reach an asymptote with these species on these inselbergs, and we also used generalized linear mixed models to determine whether the terrain, basically the slopes or the summits, and the size and the nearest neighbor distance of these inselbergs have an influence on the alpha diversity, the amount of species that can be found on these inselbergs. We also did a Permanova and non-metric multi-dimensional scaling to calculate the beta diversity using Bray-Curtis. And we visualized it with non-metric multi-dimensional scaling just to visualize how these different sizes change over different inselbergs or the different transects. So then we found, using species richness, we had 322 different species, and where you see the blue curve is the total amount of species we found on the inselbergs as well as the surrounding areas, and the pink curve you see at the bottom is when you take out the inselbergs from the data and you only sample the plains. We found 14% less species. So we'll come to that now. However, the alpha diversity, the amount of species on these transects, did not significantly differ between the sites. So for your slopes, summit, and plains had plus minus the same amount of species. However, the change might be different. And the same with the nearest neighbor and the sizes of these inselbergs, as can be seen on the right. Well, there was no significant difference between these, between the sizes or the distance that they are apart. However, the species turnover was quite significant. As you can see here, with the Bray-Curtis Bray-Curtis index there at the bottom or at the top, where that 0.64 is the percentage of difference between your summit and your plain. So your most impressive difference was on the southern plains, where it was 70% different than all the other regions, and the summits were 40% different than the slopes, and then again 70% to the surrounding areas. So that gives credit to the further up you go on these inselbergs, the more different these communities are, the structures are, and the species you'll find there. And so what does this mean? This means that although these inselbergs can't be biodiversity hotspots, as they have the same amount of species as the surrounding matrix or surrounding grassland, however, they are critically important for conservation, as they do support unique species not found in the surrounding areas, as the species turnover is quite significant. So in conclusion, inselbergs do provide a source of heterogeneity within these grassland systems, and can support different species than the surrounding area due to their environmental and their microhabitat and microclimate that's found on these inselbergs that differ from the surrounding area. Thus, it is important to focus conservation on these inselbergs, which would be actually quite easy to accomplish, as you can design your cities and agricultural use around them, and they're not that used for agricultural use and usually less disturbed than the surrounding areas. Thank you. Any questions?
Yes.
I just want to make sure that I heard you correctly. You said 15,000
Individuals.
individuals. So
Yeah.
you came with them to the lab?
Sorry?
You came with those 15 insects to the lab? Or
Yes.
what did you do with them?
So then we just ID them under the microscope, divided them into morphospecies, and then stored them away for other studies if there is any other studies.
So they're dead.
Yeah, they did. So yeah, sorry, they euthanized using ethyl acetate, and then we stored them in ethanol.
Okay. So how reliable are these
Morphospecies.
Yeah, instead of molecular weight?
So quite reliable. For each morphospecies, I took a picture as seen on the on that slide, where I used what I used as a reference individual. So every other individual you passed, I could reference them according to these morphospecies. There might be slight differences in between people, like you'll maybe get, instead of 322, maybe 320 or 24, but that's not significantly different between individuals, and we did double check it with entomologists for family level identification.
Any other questions? Yes?
You said these inselbergs provide landscape advantages, so what about them is exactly different that makes them host these different kinds of species that you don't get in surrounding areas?
So yeah, you get very different types of inselbergs. So you get your mesas, which is your flat top inselbergs made out of usually sandstone in the Free State, and then the scorpies, which is these, or nubbins as they're also known, which is this rounded dolerite outcrops. And due to these rocky outcrops, the soil is usually more shallow, and that's another thing I forgot to tell you in the presentation. So in xeric conditions, so in dry conditions, these inselbergs are usually more wetter than the surrounding area, and in wetter conditions, there are more xeric conditions, so dry conditions on these inselbergs. As for instance, if you find them in the Amazon rainforest, or not Amazon rainforest, in rainforest, the water will flow quite easily down. However, on drier environments, there's more capillary forces between these rocks, and these rocks provide little dams underground where water can be stored. So that's one factor. Then also the northern side. So the southern side has a bit more shade. That's where it's a bit more moisture, a bit more trees, and that also brings in environmental heterogeneity into the microclimate.
So a little bit water storage underneath, would you expect to find more subterranean species as well? Like more worms, more moles, that kind of thing?
That would depend on inselberg to inselberg, but due to the shallowness of the soil on inselbergs, you can't really dig on them. So you won't find really moles and subterranean species, rodents and stuff like that due to the shallow. However, I do think, like for instance, with the rocky outcrops, you'll also find more rocky species like dassies and other lizard species that are adapted to these rocky outcrops. However, I didn't focus on that. One day when I'm older, go and fitter, I'll go again and walk those inselbergs.
Just looking for more excuses to run to the field.
Yeah.
Any other questions? Yes?
A very funny question is, when you classify these inselbergs, when does a copy become a mountain? What is the ratios that you use, and how does that influence the diversity?
Okay, so the difference between a copy and a mountain, I got this question last week as well. The mountain is part of a mountain range, so it usually has one or more peaks, and geologically, a mountain is the rock from underneath the ground pushing up or moving up or boiling up due to volcanic actions or whatever. However, with inselbergs, it is eroding the softer soil away, and only these raw, hard outcrops remain. Where mountains are also eroded away over time. however, with inselbergs, it's only the harder outcrops remain in these flatter plateaus, like the Central Plateau of South Africa, and so that makes it actually quite hard to determine ages of these inselbergs, and in community ecology, quite difficult to say which came first, the inselberg or the environment they were found on, or the species that found on the environment. As the environment changed and dead species went to the inselbergs to just escape from the surrounding areas, which became harsher, or did these outcrops exist and they existed on these outcrops and then migrated to the area around the area. So long to short is these rocky outcrops is usually eroded, hard rock remaining after erosion, and mountains is usually geological, moving, rock out.
There's also always a controversy about this because where do you draw the line between height and elevation, but that's not always sure. There's still a debate about that.
Depends on where you start.
Oh, it's not, yeah. Right, any other questions? Yes?
Thank you for the lovely presentation. I'm curious about why did you let go of the default terms? We will not, was it something seasonally, whereby in winter you can actually avoid rain and all that, and you manage to get small mammals and other insects that are crawling? Or what happened? Was you just, from your presentation, you just said, okay, we used pitfall traps and then they got filled with water.
So that was a real struggle with the mosses, because we did it in one sampling season over the rainy season to get the most amount of insects as their numbers decrease in the winter. So literally every time I went to the field, one day it would rain, or a day, because the pitfalls have to be out for 8 days. So I can do my sweeps like 4 days now, and then it rains, and I can do it 4 days afterwards. However, the moment it rains, they get filled, and these cups just flip out of the ground, like the water falls around them, underneath them. And it was a real struggle. So every time I came back and said, okay, we have to go again and try again, and it was a particularly wet year. That always a struggle.
Right, I'll allow one more question. Do you have one?
Yes, I'm still on the 15,000. So I'm sure you're not the only one doing the inselberg. So how does this then affect the population biology of,
Like, do they decrease in
Yes, I
population?
mean, is there going to be any change of the species? I mean, 15,000 is a lot, and they're euthanized. And
50,000 is a lot of species. Set 3 months IDing them, but no, they don't influence the populations on these inselbergs quite that much. It's a standard entomological sampling method.
And it doesn't remove all the insects from the surrounding area, only a specific small area where the insects are removed, and they can come again from the surrounding area and repopulate where they were removed. So it doesn't damage the species present there.
Yeah, it just shows you how diverse this taxa is. For instance, 40 meters is 40 sweeps is about that distance in real life. And if you take the size, this was a small inselberg. If you take the size of the inselberg, I don't think I made a dent in the population sizes. Sampling every insect in that 40 meter.
yeah.
Okay.